And good afternoon, everyone, and welcome to Research Grand Rounds. I'm Dr. Kim Williams, Chair of Medicine here at University of Louisville, and we are pleased today to have Joohwan Park, who is one of our PhD scientists who actually comes to University of Louisville after doing his Bachelor of Science in Computer Science in Korea University in Seoul, and did a master's at, yes, he is an Iowa Hawkeye, where he did both the master's in computer science and a PhD as well. He was recruited to be at the University of Louisville. He's been here since 2021 as an assistant professor, from 2015, and he was promoted in 2021 to associate professor in the Department of Computer Science and Engineering here, and he is also joint appointed, or actually transferred over to the Department of Medicine. And he's working in the Brown Cancer Center in Bioinformatics Core, and he is working on developing bioinformatics and genomics methods for cancer studies. And he's going to talk to us today about big data and systems biology approaches to explore RNA regulatory networks in cancer genomics. Thank you so much for doing this, Dr. Park.
Okay,
great.
Right. Yeah, thank you so much for the wonderful introduction. And then I'm honored to be here to talk about the research I have been doing and I'm doing, and then the bioinformatics support that my core can provide. Okay, so before, like Dr. Williams said, I started from computer science, and then I'm in this bioinformatics field. So I just want to talk briefly about my background. PhD at University of Iowa. I went to a computer graphics class. I was amazed because the feedback was instant, and then it was just fascinating. So I thought that I'm going to work at like a Pixar or something. That was my goal at the time. Okay, and then the project I was working on was like a 7-dimensional scientific data analysis from linkage analysis. It's like a pedigree data, parents, and then their kids, and then we are try to target or locate the locus of the trait of interest, like eye color, disease, and etc. So I was so fascinated by it, and then I developed some a multi-dimensional data visualization tools and so on, and I thought that I'm on the right path to Pixar. But the data itself was so fascinating. I was like, oh yeah, I want to learn more about it. And then I added the statistical genetics part to my thesis. Then it added a couple more years for my PhD, but I didn't mind at the time. And then I was like, okay, so after I locate the locus of a trait of interest, what happens? So I figured out that, oh yeah, there's a more finer resolution to that approach. So that's bioinformatics using high throughput sequencing data. So I joined internal medicine, University of Iowa as a postdoc. And I learned so much about biology. That was a steep learning curve for me. But I really enjoyed it. And then I started developing algorithms to answer biological questions more efficiently. And then I moved to my second postdoc was at UCLA, microbiology, immunology, and molecular genetics department. And then that's where I focused more on alternative splicing, which is a dominant mechanism to generate protein diversity, meaning producing multiple protein isoforms from one gene. And after I joined here from CSE, Computer Science and Engineering Department, to BCC Bioinformatics Core, I have been doing algorithm development. Actually, I'm going to use my algorithm development and the visualization, and then machine learning, and then GPU computing. By nature, bioinformatics is interdisciplinary, right? So that's the main reason I transferred from computer science to here, because most of my collaborators are from medical school. I just joined about a year and a half ago, and then it's been fantastic. All right, so let's talk about big data first, okay? So big data is a broad term for large and complex data sets. It's too big that it's difficult to work with the traditional data processing approaches. Simplest example is, I don't know if it's appropriate, but for the attachment, right? we have a mechanism that we can send an attachment, but it's over certain size, you cannot send it, right? So that's just like a simplistic example I can give you. Like we have algorithm, but we cannot apply it because the data is too big. And then we have data from everywhere, right? Social media, online stores, digital images, videos, and so on. So the characteristics of big data is called the 3Vs. Volume, it's huge. Velocity, you have to be able to process it quickly, right? It's a streaming, right? Live streaming, you have to process it. And variety, it's not only text, it's an image or on video and etc. So how big is data? We create the 10 to the 18 bytes of data every day. That's a 30 terabytes per second. 90% of the data in the world today has been created in the last 2 years alone. That's what IBM says. And every 2 years information we are generating these days, that's the same amount of data that human generated from civilization until 2000. That's just amazing. Every minute of the day, we are generating 1.8 terabytes of data. Some illustration, it's a Domo, data never sleeps, right? Amazon, they sell $455,000 worth of it. I followed this since their version 2.0, and it's, I don't know, it's like a 10 times more or so. So no wonder their stock price went up. And there are so many like interesting things, like Taylor Swift songs, like 69,000 times, and Venmo users spend about $500,000 and so on. So that's what's happening around us. Data never sleeps. How about biological data? You already know that biological science is now data science. Biological data types, I'm just giving you three big categories. Medical imaging data, right? CT, MRI, X-rays, and 3D imaging, right? It's a megabytes to gigabytes. Proteomics, mass spec, about several gigabytes. And high throughput sequencing data, that's where my heart is. DNA-seq, RNA-seq, chip-seq, clip-seq, you just name it, to gigabytes to terabytes of data. So what is high throughput sequencing, right? It was named as a method of the year by Nature Method journal. And it's also known as next generation sequencing, deep sequencing, second generation sequencing. So if somebody's saying, hey, I want to work on high throughput sequencing data, or I want to work on next generation sequencing data, they're talking about the same thing. Okay, so don't get confused. Key characteristics, it's a massive amount of data, right? 1.0 terabases per run, if you're using Illumina HiSeq 4000 model. It's 70 times bigger than the Human Genome Project sequences. It's a short read. It's less than a few hundred bases per read. So for the Human Genome Project, 19 gigabases, it took 10 years, and then we spent $100 million. But with this Illumina HiSeq 4000, 1.5 terabases, about half a week, it's about $2,000. So the cost just dropped, right? So high throughput sequencing cost falling fast, right? It's a cost per human genome sequencing. So draft of the 2 human genome sequences was here, $100 million, right? And around 2007, next generation sequencers entered the market, okay? And then it's still about $10 million. It dropped dramatically there. So I started working on high throughput sequencing data around here, 2009. So whenever I meet somebody in the lab, and then they're saying, hey, you know, my project is using high throughput sequencing data. It sounded so cool and it sounded so ambitious. Okay, but now it's hovering around below like $1,000. So it's affordable. That's why there are so like a much the publicly available data out there. So before I talk about the alternative splicing that I'm working on, I want to point out some like important concept called the C-value paradox. It's the constant value paradox. Flies has about 14,000 genes. Worm has 19,000 genes. Rice has 50,000 genes, and the human about 25,000 genes. Does that mean rice is more complex than us? I don't think so. So there must be some mechanism that can explain this, right? So before I get to that, let's revisit central dogma of molecular biology. If one gene can make one mRNA, and then it can make one protein. Then rice is gonna make 50,000 proteins, and the human can make only 25,000 proteins, right? Which cannot explain it. So this is the mechanism, alternative splicing, right? So let's say that this is one gene. These boxes are for protein coding region. And then these lines are non-protein coding regions between the exons, or protein coding regions are called exons. And then the lines, the sequences between exons, protein coding regions are called introns, right? And then messenger RNAs, for the protein coding regions, so the intron is spliced out by spliceosome, right? But sometimes it cuts intron 1, intron 2 successfully. Then you have exon 1, exon 2, exon 3, and then you make this type of protein, right? But sometimes the splicing machinery recognizes here, but it misses this part and just keeps going and then recognizes here. And then the whole part, intron, exon 2, and intron get spliced out. Then you have exon 1 and exon 3 isoform. Then it generates this type of protein, right? So it's a dominant mechanism to generate protein diversity, but its regulation is not well understood. And the defects in alternative splicing contribute to a disease, like cancer. So this is the textbook, the example. So 12 exons in here, and then 14 exons in the B region, and then 33 and 2, they are mutually exclusive, meaning only one of the exons from A, only one of the exons from B, etc., can be included in the mRNA. So in theory, the possible number of transcripts or isoforms, proteins it can generate is already over 38,000 of them. Okay, so this one just shows how powerful alternative splicing can be. So importance of alternative splicing. I mean, so it's 90% of multi-exon human genes undergo alternative splicing. Important in the regulation of gene fusion, gene function, and errors in splicing is a major cause of human diseases. And these are 7 common alternative splicing. Okay, exon skipping. So we have 3 exons. The middle exon is sometimes included and sometimes skipped, right? So 2 isoforms, green, red, green, right? That's inclusion form, or green, green, because the middle one is skipped. That's the short isoform. Alternative 5' splice end, right? The alternative end, so green, red, green, or green, green. And this is a symmetric to the alternative 5' site. And then mutually exclusive, if red is included, blue is skipped. If blue is included, red is skipped. So 2 isoforms, green, red, green, and the green, blue, green. Okay, and alternative start exon, alternative last exon. And then intron retention. Intron, sometimes it's included in the transcript. Okay, so instead of a green, green, it just includes the whole intron here. And then because the intron is long, there's a higher chance to have stop codon. So it can trigger nonsense mediate decay. But throughout my talk, I'm going to focus on the exon skipping because it's most common alternative splicing events in higher eukaryotes. Okay, RNA sequencing. So you have messenger RNA, and then you make a cDNA, and then make a fragment, and then bring the adapters, and then so it's just a copy of your messenger RNA. Okay, so how can you use RNA-seq to study alternative splicing? All right, let's say that this is a gene, right? 3 exons, and first exon has only Cs. Second exon has AATT, and the last exon has a GGGGG. Okay, and then you just do the RNA-seq, right? And then this is what we got: CAA. Oh, CAA. So this one must be from the inclusion form, right? Because the inclusion form has a CCC, AATT, GGGGG, everything. Skipping form will have a CCC, GGGGG, no A or no T. So CAA, CCA, oh, it's from upstream junction. right? And then TGG, TTG is 1 T is from the middle exon and 2 G is from the downstream flanking exon, or 1 T and then 2 G from it. Okay, that's good. And then CGG or CCG is only from the short isoform, skipping isoform, right? So you count those, right? Let's say that you have a healthy tissue and then cancer tissue. You just count it if you have more inclusion junction rate or if you have more skipping junction rate, right? That's how you quantify or calculate the usage of the middle exon. Okay. How about ATT? This one must be from the target exon, right? So it's from inclusion form. So this one can support the inclusion of the target exon too. How about TAA? There's no such thing, right? So this one could be the sequencing error or contamination, right? So that's why we don't have 100% mapping rate. Okay. But here, this is meaningful read, and I'll get to it later in my talk. Okay, so you can quantify isoform and then detect the differential isoform usages. So that's the main thing we do. Okay, so visual example. So let's say that this is actually this is from actual cancer data. Okay, sample 1 was cancer and sample 2 was now, which are healthy. As you can see in the cancer sample, this middle exon is used 100% of the time, but it's 100% skipped, right? Sample 1 included the middle exon all the time, 100% usage, or percent spliced in is equals to 100%. We call it psi. Okay, and then sample 2 skip the middle exon all the time, 0% of usage, so psi is equals to 0. Okay, that's how we calculate it. And then I'm not gonna spend a lot of time here, but even though this one, the exon usage is 50% here, you have only one read from here, then likelihood is quite flat. Even though it has a peak at 50%, the confidence level is just too wide. And then if you have more read, you get a little bit higher peak. You have about 100 read, you get a sharper peak. So your confidence level goes up. Okay, so this one, I'm trying to tell you that that's why we need to sequence it deeper to get more reads that can be mapped to inclusion or skipping isoform. Right? So this is kind of computer science related thing. I'll just tell you. So let's say that this is from the annotation. Okay, this is a known transcript. The first transcript, 5 exons. Then I just make this spliced graph. I just make a graph. And then second isoform, 1, 3, 4, 5. So from the annotation, you can infer that, oh, okay, there must be a skipping event, right? So it's known, well known. Okay, then you just add the edge here from 1 to 3. Okay, so if 1 can go to 2 or 1 can go to 3, right? But that's a known fact, but when you sequence it and then map it back to human genome, you found that there are some reads that can be mapped to 3 to 5, which is skipping 4. Then we just added that to it. Then we have these 2 exon skipping events. Okay, that's how we build alternative splicing database. Okay, in the algorithm that I developed. Okay, so it's called the MAT. All right. So we developed it, and it's just like it's a hypothesis, and it's actual data, and then we are calculating the statistics between 2, right? In a nutshell, that's what it does. So let's say that this one RENLN gene, so exon 3 is sometimes skipped and sometimes included. But in sample 1, its inclusion level is 89% because you have 82 reads here and then 90 reads for the upstream and downstream junction. And for the skipping, you have only 11. But in the sample 2, you have 10 and 8, while you have 166 for the skipping, right? So this one is highly included in sample 1, right? So then we calculate each p-value and then FDR. So this is what we the illustrate. Okay, when you have this kind of data. So this is what we illustrate for the publication, right? Oh, okay. So this is the density, like a number of reads that map to upstream flanking and downstream flanking and the target exon. As you can see, we have bigger mount on the sample one, right? So the usage of exon in sample one using the program is 73%. Usage of exon in the sample two is only 5%. So this psi sample one, psi sample two, the difference here, psi sample one minus psi sample two is called the delta psi. Delta psi is 68%. And we did qRT-PCR, okay? And as you can see in the long isoform, we have a darker band, and then sample two, we have a darker band in the short isoform, skipping isoform. And when you quantify that, it's 80% versus 2%. So the delta psi is 78% there. So we validate it. And not because, the sequencing technique is so advanced nowadays. So the validation rate, when I first started this alternative splicing, it was about like 80 and 85%. But now it's about 95% or higher. So that's the first one, right? The one read, one RNA-seq per sample. That's all we have. But as you saw, the sequencing cost dropped, right? So now people started generating replicate, right? So there are like so many the sites you can go to grab publicly available data set, right? So we got a lot of emails asking, hey, I have 3 replicates, what can I do? And then at that time, what we could just tell them was just like pull them together and then run maths, right? Which is not ideal. So we started working on our maths, and then it was still like it's widely used like internationally. And then we got like over 15,000 downloads. I checked it yesterday, and then it was on PNAS and molecular cell. And then we are keep updating it. Actually, I'm not working on this anymore because I left to UCLA, right? And then my previous PI went to UPenn, so that's where he's keep developing. But this one is really good. And then RMS statistical model. I don't want to give you a headache. In a nutshell, replicate, it will tell you all different size. How can you make average size? Okay, for the sample one and sample two. Okay, that's why we use the binomial and then a normal distribution and this one. And then this part, of course, biostatistician did it. Okay. So here it shows that, okay, 3 size, right, within the constraint. But as you can see, this dot is close to these 2 triangles. It's because they have more reads. When you have more reads, it kind of goes towards that estimation. And then here is the same thing, these 2, and then this dot is not in the middle. It kind of goes to this way. And then you see the difference and then calculate the p-value. And then we report it. So how can you apply that to the actual study? Okay. So the epithelial to mesenchymal transition, EMT, right? It's they have like roles in development and the metastasis, right? So there's no such a thing like good cells and bad cells. Right? But when it goes to metastasis, and then epithelial cell is a good cell, and then mesenchymal is a bad cell because of they are evasive and then go to different organ, right? So how did it, this is the study design. Let me see how much time. Yeah. So prostate cancer cell, it's called PC3E, okay? And we inject HH from University of Iowa, the Henry lab did it. Then in the tail vein, and then we harvested the mesenchymal cell, the GS689 liver from it. And then we made 3 replicates per sample and then did RNA-seq and 1.5 billion reads. It was a huge, huge amount of data. And then we ran our maths, right? And then we found that this is one of the exon 13 of SLK gene. as you can see, you don't see any peak in mesenchymal sample, but you have inclusion in the PC3E epithelial, right? And sometimes you find a group of exons included at once and then skipped at once. Okay. So that was like a very, and then this one is like a known the variant exons that related to the cancer. So alternative, so we ran our maths on that, right? For the exon skipping events, we detected not about 93,000 skipping exons. And then of those, 557 were significant, meaning it shows enough difference between sample 1 and sample 2, epithelial and the mesenchymal, and the FDR is low enough, less than 5%. Okay, so we got 557, and 28, 26, and so on. So out of about 100,000 alternative splicing events, we found about 900 significant ones. And then, first of all, we have to check, are they real, right? So we did the validation. And then I don't remember it precisely, but it was over 95% validation. So it was good, validated, right? And then once we know they are real, what do you want to do? What do they do, right? That's the next question. So we just did the famous gene ontology co-enrichment analysis of using 557 exon skipping events, right? And then these are biological process, molecular functions, and then cell components. So, okay, that's good. So we know they are real, and then we know what they do, okay? What is the next question? Who's controlling it? Who is the driver, okay? So we wanted to see that what's making this different alternative splicing, okay, in epithelial sample and the mesenchymal. So we examined RNA binding proteins and then splicing factors, okay, and then compared their expression levels in epithelial and the mesenchymal, right? And then we found so many familiar genes there, okay? For example, ESRP1, muscle blind like 3, NOVA, and the RBFOX2. And we were kind of surprised that QKI, King, was not differentially expressed between those two, because we expected to see the differential expression of QKI, but it didn't. But that's what data said, and then we just accepted it. So we examined, okay, so of those 557, are they known to be the target of any splicing vectors? And then yes, yeah, ESRP, they have 85, about 90, okay? The exons were known as ESRP1 targets and RBFOX2 about 40 something, and then NOVA about 10 of them. So it's like, oh yeah, it must be ESRP then. I mean, it has a higher chance, right? That ESRP is the driver of that splicing difference. So ESRP means epithelial splicing regulatory protein 1. So, okay, so it is talking about the epithelial. So that's good. So what can he do? If it's the driver, what can he examine that if actually doing some functions there? So, okay, to be able to work, it should be bind to the mRNA and then control the splicing, okay? So we examine the motif of the ESRP motifs. So there are 12 of them, GT rich, like UGG, or UUGGT, things like that. So we collected 12 of them and then examined all 557 exon skipping event exons, and like by upregulated ones and then downregulated ones. And then of course we need some background too. So we used, we randomly collected about 2,000 exons that didn't show differential usages between epithelial and the mesenchymal. And then this is what we had. So, okay, so this is the target exon, right? And this is the downstream intron. As you can see, the red one, epithelial specific, has a lot higher, higher frequencies of the motif, right? Compared to background or mesenchymal specific exons. But on the contrary, for the exon body itself and then upstream intron, you have a higher peak for the mesenchymal, okay? So, oh, okay, that's what we saw. And then we did some literature search, and then that's known fact. So that means the ESRP is working as it is supposed to. So when ESRP binds to the downstream intron, it promotes the inclusion of the target exon. Okay, so the theory behind it is like it's binding there, and then slows down the spliceosome, and then making sure that it recognizes this splice site. Okay, and if it binds to the exon itself or upstream intron, it's covering the splice site. So the spliceosome cuts here and then failed to recognize here and then just to cut here so that this one is skipped. Okay. So this one matches that, and then it's like, oh, okay, this is a bioinformatics approach, right? If you can examine one splicing factor or RBP, we can examine anything with no motif, right? So that's how we developed RMX1 and RMX2. And then we have author there, Jay Huang. He just worked hard to make it. And then we have it published in 2016 and 2020. We are still getting a lot of emails and then inquiries and then access. So we are now, it's hosted here at UofL. Okay. And then we are thinking about making RMX3. But I don't know when it's gonna be out, but that's in my to-do list. Okay, so that's about the alternative splicing. But after we have alternative splicing and then RNA binding protein, right? So we wanted to see more. Okay, and then when I was at the UCLA, there was some guest speaker who talked about circular RNA, and I thought, wow, that's a really interesting concept. I want to work on it, and then. Luckily, I was involved in circular RNA project on the first year I joined here at UofL. So the concept of the circular RNA is like this. So for the canonical splicing, it just splice out all introns, and then the isoform that you're gonna make is exon 1, 2, 3, 4, right? But sometimes it just cuts here, and then somehow end of the 3' of the exon 3, and then 5' of the exon 2 join together and then make this circular form. Okay, so how can it happen? So one of the like a biogenesis of circular RNAs, like a known one, is it's a reverse complementary allele sequences in the introns. Okay, and then they bind together and then make this a circular form. Or sometimes the RBP promotes the circularization of the transcript. So it can make linear form, and at the same time, it can make circular form that contains only exons. It's called exonic circular RNA, or sometimes exon and the intron. Okay, so that's one of the like well-known biogenesis of circular RNA. So circular RNA research in my lab, right? You first want to detect the circular RNA, right? So we detected it. And, the machine learning is one of the my research topics. And so we wanted to see that if we can apply a deep learning approach to classify circular RNAs from other long non-coding RNA. And then we published that paper too. And then the current project, our main focus of the circular RNA is the translation of the circular RNA. So circular RNA was known to be non-coding for about 20 years. Okay, and then people never doubted, oh, okay, it's a non-coding RNA. It's doing some important things like microRNA sponges, okay? But they didn't think that it gets translated, okay? So what is a microRNA sponges? So circular RNA, circular form, right? It doesn't have a 5' or 3' end. So it's supposed to be resistant to what is it called?
Exonuclease.
Huh?
Exonuclease.
Yeah. So it's just supposed to be resistant to the cell decay, right? So it's supposed to. So some studies are saying that it can last about like 2 weeks. Okay, this is really, really long time, right? When you compare it to a linear RNA. The expression level of a circular RNA, right? When you have like 100 million reads, we are getting less than 100 million reads from circular RNA. So each expression level is, we usually call it 0.1% of the total reads are from circular RNA. So it's a really small portion, but it does important function because it can last longer. Okay. Yeah. So oh yeah, now I remember it. So it's resistant to the degradation process. That's what I meant to say. Okay, so it's there, and then if it has a microRNA binding site, right? MicroRNA should go to the 3' of the linear RNA and then suppress, right? Its function. But microRNA sees the same binding site and it's there for about 2 weeks, then it just binds to there. So if we need the microRNA function to stay healthy, this circular RNA can mess up that process, right? So that's like a well-known, most important circular RNA functions, microRNA sponges. Okay, and then recent studies provided strong evidence for the translation of the circular RNAs, right? So when they found the translation of the circular RNA for the first time, just finding one translated circular RNA was big enough to publish in Molecular Cell. Okay. And then there was a back-to-back submission, and then there was big news for circular RNA study group. Okay, and then of course the proteins translated from circular RNAs are novel because the sequence doesn't exist in human genome. But when it's circularized, there's some sequences that goes over here, right? 3 to 2, right? That's a novel sequence. Okay, so that's why the the protein translated from circular RNA is novel. Okay, so it can promote or suppress the tumor formation. It is a known function, and then activating molecular pathways related to cancer development. Okay, so alternative splicing and circular RNA are associated with various diseases. The defects in AS are responsible for numerous diseases, including cancer. And the abundance of circular RNA has been highly correlated with colorectal and the ovarian cancer proliferation. And circular RNA that functions as a microRNA 7 regulator is less abundant in Alzheimer's disease samples. So this is just only like 3 examples, of alternative splicing and circular RNAs associated with disease, but you can just find like unlimited cases. Okay, so how can you detect translation of circular RNA? Okay, so RNA-seq, right? The secret paper that I published that will detect circular RNA. Okay, so it detects it. And ribosome is the sequence of the fragment covered by ribosome. So that means it was being translated at the moment of sequencing. So that's the evidence of the translation. Does it make sense? Okay. All right. So let's say we detected these circular RNA. Okay. So this is exon start, and then it's exon end. It's one exon circular RNA. And then when it meets, it's called back-spliced junction. Back-spliced junction. So we are getting 50 BP here and then 50 BP here. And then exon 2 and exon 3, back-spliced junction, we're getting it, and then we put it in some circular RNA specific database so that we can map it. So we map reads, RNA-seq, we map ribose-seq reads to human genome first. Okay, and then there should be some reads that cannot be mapped. And then we don't know where they are from, right? And then we map that back to this database, circular RNA database that we just developed. Then see that these blue ones are the individual ribose-seq reads. And then if it goes across, mapped across back-spliced junction, then that's the evidence of the translation of a circular RNA. And then they should show 3 nucleotide stride, because that's the size of amino acid. right? If it uses 3 nucleotide, and then you got these read, and then, yep, that's the evidence that we can find. And then we can apply this to existing RNA-seq and the ribose-seq pair that's deposited to an NCBI GEO or things like that. And we have house the data from rat, right, Jay? Right, and then how many did we find that kind of case? Only 1 or 2, right?
Yes.
Yeah, I mean, it's really rare. It's really rare, but when you find it and when you can validate it, it's a huge discovery. And then we want to, actually, we're almost done developing this automated pipeline. Okay, you just give RNA-seq and the ribose-seq, and then we'll report these are the circular RNAs, and these are the ones that might get translated. Okay, so that we are wrapping up that pipeline, and then we're gonna submit that paper too. So how can you validate it from WellLab? Circular RNA, the translation of the circular RNA. So let's say that this is the circular RNA exons. Then we found that these get translated, and then we just put GFP protein, the partially here and then here. If you get circularized, we'll have exons and then GFP in the correct order, right? And then you're gonna see the green light. Okay, so this is how you can validate it, and then it's a Minigen reporter on validation. Okay, so summary. So the era of big data introduces new challenges and opportunities, right? More like opportunities for me. And then, we cannot function as professionals unless we are comfortable with dealing with the big data. That's a hard truth. RMS is a statistical framework to handle or quantify the analysis of alternative splicing using the big RNA-seq data. And then using RMS and the RNA-seq, we revealed the important role of a splicing factor ESRP1 in controlling metastasis associated with splicing regulatory network. And then circular RNA functions such as both non-coding and then coding RNA. So data is getting bigger, right? And then accessing and handling the data is a big challenge. I mean, even getting a data from other PIs is sometimes challenging. If it's in the basis space, it's easy. We can just download it. If not, then if it's in their local computer, we recently went through that process, and then instead of taking only half a day, it took more than 2 weeks. So she has to download it to the USB drive and then give it to us. We transfer back and then transfer back to the cluster. It was a long process. Okay, so what Bioinformatics core can do for you? So we can do a consultation, right? And then say, okay, this is your target. Okay, this is the sequencing techniques you can apply to your project to achieve your goal. Gene profiling, gene ontology term, enrichment analysis, pathway analysis, alternative splicing, RNA binding protein motif, and then sequencing we work with are like bulk RNA-seq, single cell RNA-seq, of course, and then 16S, ChIP-seq, CLIP-seq, nanopore, PacBio, you just name it. Okay, if we never worked with it, we'll find a way. Okay, so this is I just put it here to give you how the process will go through once you have a really good research idea. Okay, you just contact us, right? We're gonna do the consultation. This is the ideal process, by the way. And then based on our the discussion, you can we can choose some sequencing, and then sample prep, and then sequencing, right? You can use here. We have a sequencing center here, or you can outsource it. And data analysis, right? So sometimes we start from here. I mean, when I say we, it's a Bioinformatics core here, but sometimes you have all the data set, then we start from here. Okay, so actually we can jump in anywhere. Okay, but it's either consultation or data analysis. That's from my experience. So when you do the data analysis, and if you have some lab member who wants to learn, we can train them. Right? These are perfect example, right? And then, there's a result, we share it, and you can use that for the publication or you write grant, right? That's the whole process, an ideal process. So when you have an idea, just reach out to me. Just send an email. Oh, I have a great idea. Can you have a short meeting there, MS Teams or something, right? So and then some other projects that I want to mention. Okay, I'm not going to go in detail. The GPU accelerated computing. You heard so much about GPU. It's not for the cryptocurrency. It's for the research, right? And then on cloud computing, right? I had to move data from Iowa to UCLA, UCLA to here, computer science to here, right? It took at least about 2 months per transition. So if you could put everything in the cloud computing, then I didn't have to worry about, moving data around. Okay, this is probably the last one that I want to talk about, the time course data analysis. Sequencing cost is so cheap, right? And then it's not like binary. You have one sample and then the other sample, control and the treatment. Sometimes you have control and then you treat it and then you sequence at various time points. You can do it because it got cheaper, right? And then you can make a lot of replicates. So this is one example, okay? Z1, mCherry, it's known to trigger EMT, okay? So it's a doxycycline treated and then with like a EMT, like mCherry expression is getting higher, and then the EMT happens from 0 to 7 days. ESRP, which is epithelial specific, its expression level goes down. Same thing for the E-cadherin. And then vimentin, which is a mesenchymal specific, its expression goes up. And then this one is like a beta-actin, right? Housekeeping gene, right? Just shows it. So, okay, this one works great. And then we got 3 replicates per time point. That's 8 times 3, 24 time point. And then this is what it says. So this is the group of gene that gradually goes up, right? From blue to red. And then transition up, it goes up and then goes down. Transitions down, it was high at the beginning, low in the middle, and then go up again in the later days. The down, it's very high at the beginning, it's low at the end of the experiment. Okay, so we have, I don't know, some a handful time series data, and then the algorithm to efficiently compare the time points and then classify the genes based on up, down, transient up, transient down, or there might be something between. Okay, so those the algorithm to detect or handle the time course data analysis is still underway. Right. So that should conclude my talk, and I would like to thank everybody in the bioinformatics group, especially Jay and Sanhua, and our external collaborator, and then here, I have such a good friend and collaborator, Levi, and then Leah, and then so if you haven't tried bioinformatics approach yet, this is fascinating, and then don't get scared. Okay, so it's okay. It's okay. You generate it, and then we'll help you. Or even before you generate it, we can, have a short meeting to see that if it's feasible or not. Okay, all right. Thank you so much, and then you can spend it.
Open for questions.
Right.
So the circular RNAs, so you said they have identified some translated.
Yes.
And you guys have also identified, so.
We haven't validated it.
Okay.
Yet.
So, but the ones that have been identified?
Yes. They used to do some mini-gene reporter, and then they showed it.
So do they look like the protein, the messages that came from a little bit? Or is it, because it depends on how I'm just trying to picture in my head how it gets translated from the circle and what frame it would be in.
Yeah, that's a good, it's iris, internal. Yeah. So you just identify from the second line, that starts from there.
Okay.
Yeah.
All right. So does it have a different amino acid sequence in? Because the frame might be different than the gene that it came from that it's supposed to be translated.
Right, and then one a kind of interesting thing about that is sometimes it's divisible by 3, then you make the same amino acid.
Yes.
But if it's not, on the second time the ribosome goes the different amino acid and then different amino acid, unless it has a stop codon somewhere. Yeah. So sometimes it does 2 circles and then stop codon, right?
Oh, yeah.
Yeah. So that's very, very interesting.
So how big is usually the circular RNA?
Very good question. Yeah, because a lot of people want to know that. So it cannot be short because it should be able to bend and so on. So in average, it's 300 BP.
Very small.
Is it? Okay, I thought it's
Very small, and they cannot imagine all these.
no, there are long ones too, of course. There are long ones too.
Okay.
But when you make the histogram showing the distribution, the 300 has a peak.
Okay.
Right, kind of
So
tails
most
off.
of them are
That's right.
300.
That's right. And then most of them have 2 or 3 X.
So I just assume you based on the only 300 base pair, and the translational machinery won't be able to build up on such a small molecule.
I don't know. I know the exact answer to that, but the 2 translated circular RNAs reported were long ones, like 15,000.
So that's why I can imagine. Okay, it's not physically available, for the whole entire translational machinery built on such a small molecule.
Yeah. So if we find like a translated circular RNA with only like a 300 BP or even 500 BP, you think it's a big discovery?
No.
No. I would be interested in that.
Yeah, for me, I don't know, okay, but I just kind of imagine all these proteins must be built on such a small molecule.
Okay.
I don't think it matters the size, it matters what the protein that it makes does in the end, right?
Yeah.
Because even if it's 300, right, that's 100, but if it goes around.
Yeah, it goes
times,
around and
you
around.
could get
make
a couple hundred amino acid protein.
But I say that the translational machinery should be, individually working all these proteins together to finish the job. But anyway, okay, it's all right. By the way, also I would like to know how now you say it's cheaper for the deep sequence or whatever. How cheap it is for a regular sample, okay?
So you're talking about bulk RNA-seq or?
Yeah, as RNA-seq. Of course, depends on how deep.
Right. I think 460 million reads, right? And then if you have like 3 replicates and 2 samples, it's how much is by 8? 60 million each, right? Per 60 million reads per replicate. The last time I saw was, I might be wrong, but about $3,000 for sequencing only, but library prep fee is added per replicate, which is about $200.
Okay.
Yeah.
So,
but if you contact sequencing technology center here, they can give you more.
Melissa, right?
Yeah, Melissa stepped down, so it's Nick now or Elizabeth.
It's so dynamic. Okay.
Yeah, she can give you like accurate estimate.
I have the name. Okay, good. Yeah, I want to know. Then I can see how much really need for me to finish my job.
Yeah.
Otherwise, as you suggest, maybe we pull the sample together. Or we have to do, multiple replicates.
Right. Yeah.
Fantastic.
Right.
Thank you so
Thank
much.
you.